Welcome to the notes video on chapter 2.5, which is on enzymes. And you might as well get cozy now because this is one of those topics that bridges almost every other topic in the whole IB biology course. Now, we have hundreds, thousands of different metabolic reactions that have to happen in our cells in order to sustain life. And the sucky part about most of them is that they're really slow. And if we let them kind of do their slow thing on their own, then they would take place at such a pace that life wouldn't be sustainable. Okay, so to speed them up, we can add something called a catalyst. And a catalyst is any substance that increases the rate, so it makes things faster without being consumed. So it doesn't get used up in the process. An example of a biological catalyst is an enzyme. Enzymes are globular proteins, and that means that they have tertiary structure. And they speed up biological reactions. So here's kind of a look at a tertiary structure of an enzyme. I can see these alpha helices, I see these beta pleated sheets, and they're all kind of just folded together in a really specific shape. Oh, and I guess I gave away the answer to this third question. My bad. Tertiary. So this specific shape that enzymes have determines their specificity, So enzymes have a very particular shape, and that shape is determined, again, by the amino acid sequence, because the amino acid sequence, that primary structure, helps to determine all of the other levels of protein structure. The way that those bonds and those R groups interact determines its exact shape. And of course, this is determined by genes, So your genes control the shape of your proteins. Remember, through that transcription and translation process at the ribosomes, And that all leads to a very specific enzyme with a super specific function and shape. Now, why is that shape so important? Well, that's because it has to have a very particular active site. And the active site is the spot on an enzyme where the substrate attaches. Substrate is just another name for the reactant in an enzyme catalyzed reaction. So for example, in order to break down the lactose that's in milk, into its primary monosaccharide components of glucose and galactose, you need to add in an enzyme called lactase. Lactase has a very specific active site that will only fit lactose. So you need one enzyme per substrate that you're working with. Lactose fits in perfectly to this active site on lactase. Now, there are a couple ideas in terms of how exactly this substrate fits into the active site. And kind of the old school model, the one I learned when I was your age, is called the lock and key model. And basically that says that only one substrate can bond to an enzyme, and that's because the substrate is a perfect match for the shape of the active site. That had to mean that the active site was a rigid, no negotiation, not changing my shape kind of place on the enzyme, right? Well, now we realize that that's really kind of old school, So kind of the more modified, the more scientifically accepted version of how enzymes interact is called the induced fit model, And I always think of this word induced with pregnant ladies, because pregnant ladies whose babies are too cozy and don't want to come out, often their labor gets induced. We inject them with drugs to make them have that baby. Well, here we are making that substrate fit in with the enzyme. It is important to know that each enzyme can only act on one substrate. Lactase can only work on lactose. Okay? Amylase can only work on starch. Okay? Each enzyme can only work on one thing. But we now know that the shapes of enzymes do funky things when their active sites get filled by these substrates. Okay? So what's happening here is when the substrate binds to this active site, it kind of causes an overall change in the shape of the enzyme, kind of like a gloved hand, like fitting around something that it's holding, right? Okay, it changes its shape once something is in it. Now, enzymes are pretty cool, but they're not magic, right? So it's important to remember that enzymes are only going to help reactions that would have taken place anyways, okay? So I cannot make lactose break down into, well, it'd be helpful if I could spell this right. Okay, it won't help lactose break down into like money and smiley faces. Okay, so things that aren't going to happen normally aren't going to happen with enzymes. But things that do happen, but they're just slow, can really be helped out by enzymes. And what they do is they lower the amount of what we call activation energy. Activation energy is the energy that you have to put into a reaction to get it started. Remember that molecules need to collide with each other in a specific way with a specific amount of energy in order to produce a chemical reaction. Well, what enzymes do is they reduce the amount of energy that you need in order for that collision to result in a reaction by placing those molecules in a favorable position and kind of weakening their bonds, okay? So they make sure that those molecules collide at the right angles, in the right place, so that you don't need as much energy in order to get them to react. Now, they do not provide any energy. They're not used up, and they wouldn't help reactions that wouldn't occur normally. All they do is they lower the amount of energy that you need in order to get something started to begin with. So as we talk about factors that can influence how enzymes work, it's important to know that, yeah, there are certainly factors that have nothing to do with enzymes that can still affect reaction rates, okay? So it's important to understand that there are factors in the environment that can affect enzymes for sure, but there are also other factors in the environment that can affect reactions as a whole. Okay, so that could be like what kind of substrates are available, the temperature, all kinds of things. Okay, let's talk about this effective temperature. We talked about this a little bit when we were talking about proteins. Up here is what we call the optimal temperature, and I can tell it's the optimal temperature because it's the temperature at which I'm getting the maximum amount of activity out of my enzyme. So again, you don't really need to worry about these axes headings. You just need to know that at an enzyme's optimal temperature, that's the temperature at which that enzyme is working the best and the reaction is going fastest. So I could have also said over here that this is the rate of reaction. All right. Up here in this part of the graph, we're seeing a steep drop off, and that's because the enzyme is being denatured. Okay, so remember, enzymes are proteins. They are really susceptible to changes in temperature and pH. Okay, and when you expose them to high temperatures, you're causing that protein to become unfolded. You're breaking those hydrogen bonds, and now it doesn't have the right shape anymore. Down here, okay, this is having nothing to do with the enzyme, but cold temperatures, okay, make molecules move slower. Okay, and when molecules move slower, reactions don't happen at as great of speed. Okay, that's something that has nothing to do with enzymes. Okay, same thing here with pH. So we've got, on this time, pH on our x-axis. Okay, and again, rate or maximum activity, doesn't matter, those are synonymous, over here on our y. This tippy top of our graph is the optimal pH. So this is the pH that our enzyme is happiest at and working the best at. Okay, up here in these high pHs, okay, these are bases, and in here the hydroxide ions from the different bases, okay, are denaturing the proteins. So hydroxide ions from bases are denaturing the enzyme. So it's ruining permanently the shape of that enzyme. Over here, same thing. Okay, but this time it's hydrogen ions from acids are denaturing the enzyme. Okay, and again, denaturation is a permanent change. Those enzymes are never ever going to work again. Now the last one is a little bit trickier, okay? And so this plots the effect of substrate concentration on enzyme activity. All right, let's say that this is my enzyme and this is my substrate. If I have this substrate, okay, in solution with my enzyme, this molecule may have to bounce around for a while before it bounces directly onto the enzyme's active site. Okay, so if I only have one of these substrates in my solution, I'm probably gonna have a really slow reaction rate because it might take a while for that substrate to kind of like come in contact with my enzyme. As I start to add more substrates, okay, the possibility of these substrates bouncing onto the enzyme become greater and greater. First of all, because there's more of them, so there's more bouncing around. Second of all, they might be bouncing off each other and then onto here. They're running into a lot of stuff, and they're going to be bouncing into this enzyme with greater frequency. And so that's going to increase the rate of the reaction. Okay, so if we start labeling things down here, okay, here we have not very many substrate molecules. Attaching to the active site. Okay, as we increase that concentration, okay, then what we're seeing here is substrates bind with the active site more frequently. All right, and then finally we get this plateau shape, okay? And here we can say the rate plateaus or levels off because all enzymes are busy with substrates. Okay, and adding any more won't increase the rate. Okay, so this enzyme can only handle one substrate at a time. Okay, so if it's busy, it doesn't matter how many more of these things I add, okay, it can only catalyze one reaction at a time. So adding more and more and more and more and more and more and more isn't going to be helpful, and I'm going to see this rate plateau. Now, you've heard me mention optimal temperatures a few times. Okay, optimal temperatures are very different for different enzymes, and that's largely dependent on what kind of organism it's in. So if you are a human, and humans have a natural body temperature of around 37 degrees Celsius, it's likely that your enzymes are going to have an optimal temperature that's somewhere around the same as your body temperature. Okay, they've evolved to have proteins or enzymes that work best at those temperatures. If you're an organism like some kind of a bacteria that lives in a hot spring, your enzymes have probably evolved to be most functional at a temperature in which you live and that your cells are at. Okay, so every cell has different optimal temperatures, or every enzyme has different optimal temperatures. This human enzyme and this bacteria enzyme are similar, but they're two different versions. And because they are slightly different shaped, they have slightly different optimal temperatures. Okay, they're going to work in different environments. No matter which environment we're in, we're still seeing the same shape. Slow reactions because of low temperatures, optimal temperature, and then a sharp drop off due to denaturation. The same phenomenon happens with pH. So let's look at a couple examples here. Okay, you have in your saliva an enzyme called amylase. You're going to hear that about a good jillion more times. Amylase breaks down starch. Okay, and its optimal pH is around 7, which, gee golly whiz, is about the pH of your saliva. You have an enzyme called arginase, which is produced by your liver, and it is kind of secreted with this stuff called bile, and bile has a really basic pH. Okay, so because that enzyme is hanging out in that solution, okay, it has an optimal temperature of that pH. Okay, if I try to put it in a pH of 7, it does not work very well. It gets denatured. Same thing on the other end of my scale. Pepsin is an enzyme that breaks down proteins, and it's found in your stomach. Your stomach has a really low pH. Okay, this enzyme just so happens to have an optimal pH of around 1 to 2, okay, which is the same pH as your stomach. If I put it in a pH of 7, it starts to get denatured. So every enzyme has its own optimal temperature and its own optimal pH. Now, we can recognize the importance of enzymes in our bodies, right, to speed up these metabolic reactions that have to take place. But what about in industry? Okay, well, a lot of times the medicines that we take are actually enzymes, and a lot of the foods that we eat are produced using enzymes. But getting these enzymes out of cells is freaking hard. It's almost impossible because they're usually, like everything else, dissolved in solution. So it's not like I can put them through a filter or just like ask them nicely to come to the side. Okay, they're kind of a pain in the butt. So to cope with this problem, we put them through a process called immobilization, kind of like exactly what it sounds like. They can't move. So immobilization is an industrial process that traps the enzymes in chemical pores. So we kind of put this chemical in there that has certain parts to it that are going to attach to that enzyme. And it bonds with that enzyme and immobilizes them. They're now stuck in this bead-like structure. Okay, so they become immobilized. We can actually pull them out of solution. So imagine, and this doesn't work. I mean, you can try it if you're dumb, but imagine if this did work, it'd be so cool. Sticking a magnet into a glass of salt water and having all of the sodium or chloride ions come out of solution and stick into that magnet. That would be awesome. Okay, well, that's exactly what's happening with these immobilized enzymes. We can use those immobilized enzymes for lots of important things, okay? One of them is for producing lactose-free milk. Well, let's go back and review a little bit about lactose. Okay, lactose is a disaccharide Okay, so made of 2 monosaccharides or sugars. It's a carbohydrate, and it's found in the milk of mammals. All mammals produce milk. Milk comes from mammary glands. That's how we get the word mammals. Now, most people in North America, where we live, are lactose tolerant, but really in the world, there are far greater numbers of people that are lactose intolerant. We're going to look at the evolution of this lactose tolerance sometimes in class, but it's a really interesting phenomenon to take a look at. The reason is because we're not supposed to be drinking milk as adults. You see, you should only be breastfeeding when you're little. So what happens is you have this gene, okay, part of your DNA, which codes for the proteins to make the enzyme lactase. When you are little, that gene is turned on because you need this enzyme lactase to break down the lactose in your mother's milk. When you're older, you shouldn't be breastfeeding anymore. If you do, that's strange. Don't tell anybody about it. Okay, and this gene turns off. So in normal people, this gene turns off around the time of puberty, which to me is still too late, but whatever, no one asked me. Okay, and most people aren't able to break down lactose anymore. About 10,000 years ago, a mutation occurred that allowed this gene to remain on, okay? So they were able to continue to produce lactase well into adulthood, and then those people ended up migrating to places like Europe and North America. And so we see these pockets of people who are lactose tolerant, but most of the world is lactose intolerant. Now, these poor lactose intolerant people still want to have good lives. They still want to be able to eat cheese and eat ice cream and drink milk and eat yogurt and do all those things. But when they do that, they lose a lot of friends because they have some bad digestive problems. Okay, and here's why. When that lactose is in your small intestines, okay, it can't make its way through the holes in your cells. It's too big, okay, to be absorbed into your cells. That process doesn't happen. So instead, it stays in your small intestine. Well, down in your small and large intestine, there are all kinds of bacteria. And these bacteria think lactose, the sugar, is super duper yummy. And they eat it and they produce gas. And that gas gets trapped inside of your digestive tract, causing you to have bad cramping. Your body doesn't like the way that feels, so it tries to poop that out as fast as possible, and you get really bad diarrhea. Well, what's supposed to happen? Okay, if you can produce lactase, it takes this lactose and it breaks it down into its primary components of glucose and galactose. So it breaks them down into its 2 smaller sugars. These can pass through the holes, okay, they're small enough to diffuse into the cells. So they don't make it down here into your small intestine. There's no more sugar left. These bacteria are sad, but now they don't have anything to eat and cause you all these problems. So how can we give these lactose intolerant people, the people who don't produce any lactase, a chance at living a full and wonderful life of dairy products? Well, it's pretty simple. You take milk and you pour it through some of those beads containing the immobilized lactase. As the milk makes its way through this lactase funnel, the lactase breaks the lactose into its 2 primary components of glucose and galactose. Okay, so then we end up getting lactose-free milk. This lactose-free milk, instead of having lactase, has glucose and galactose. So it doesn't taste disgusting, okay? It's just a different form of the sugar. We've just broken that sugar down for you. Okay, and so for all of you that are lactose intolerant, I thank you for drinking this stuff and not the regular stuff before you come to class. And I will see you in class. That'll do it for chapter 2.5 on enzymes.